Can we simulate blindsight in monkeys? So to understand the neural base of blindsight, we really need to understand what's happening. We need to have a model animals. And in fact, the blindsight can arise when the primary visual cortex B1, which I'm going to explain this week and next week, is selectively damaged in monkey. And without V1, primary visual cortex, monkeys can perform various visually guided tasks accurately, but they seem to be blind, like blindsight patients. All right, so here, this is a little bit of a review of what the task in the localization versus detection task in the blindsight subject GY. So here, if you focus on this red arrow part, this means that the participants GY, blindsight patient, upon seeing 46% really high contrast stimuli, he said that roughly like 20% or of the time that he did not see anything. So he denied seeing things. However, roughly 80% of the time, 80% of the time, he was able to correctly localize when the stimulus was either here or there. Okay, so the performance is chance at 50%, but his performance was 80%. And as you saw, the masked stimuli in the Dehaen's paradigm, performance itself was almost chance, right? When people reported not seeing. So this is quite different from the masked situation, okay? And this is a really good reason to start doubting what the patient is actually reporting. In the end, we don't know exactly how they are feeling. So we need to be careful about interpreting what the blindsight patients are reporting. Okay. So before dismissing, we just need a stronger evidence of the blindsight. And to back up that phenomenon, let's study the primate model of the blindsight. That's the motivation. And the primate model is quite advantageous in various ways. One is that the spatial temporal resolution of the brain lesions. So unlike the blindsight patient, researcher can carefree and also targeted manner in terms of the spatial location boundary of the lesion and also timing of the lesions. In the case of the blindsight, then we don't know whether there is some kind of plasticity happening, and we also don't know when exactly this kind of lesion happened. So that's something that we can control with the monkey model. And also, there's no social incentive to tell a lie. If you are skeptical, then you might think that oh, maybe blindsight patients are actually seeing things. But because they can become famous, if they act really weird, then they just behave as experimental want. But such a communication or social incentive is impossible for monkeys, presumably. So that's a good way to check that. And also possible to perform invasive experiments to better understand neural mechanism. Of course, in the case of the monkeys, we can insert electrodes to record the neurons eventually, or manipulate neural circuits and so on to better understand the neural mechanism underlying the blindsight, to better understand what happens in the human brains, and also possible ways to cure the disease, or including blindsight in the future. But of course, there is also disadvantage, and the most prominent is a lack of the power of communication. So that means that we need to establish some kind of ways to understand what they are experiencing in many different ways. And also training is sometimes required when the task becomes very, very complicated. And training itself actually changes the brain circuits. So that renders this advantage of temporal resolution to be a bit, kind of, disadvantageous. All right. So now let's look at this video of Helen again. But paying attention to this particular part, 5 minutes and 37 seconds. Okay, so this is a video of the Helen in the longer version and around 5 minutes. And here background, there is an ongoing noise sound, and so that Helen cannot use the sound echolocation to avoid obstacles. And let's see how it goes. So she's exploring, and then here she hit on this transparent wall. Okay, and but she doesn't actually hit on the other transparent wall, as long as they have a high contrast stripe patterns. So that's important thing. And if you're interested, please watch the rest of the video to see what the Helen's behavior is like. It's quite interesting. Okay, so as you saw, under the white noise masking condition, she couldn't actually use the echolocation to avoid obstacle, and then hit onto this low contrast transparent wall. And that is consistent with the behavior of the blindsight patient, where they couldn't actually see low contrast stimulus. For example, here, when the contrast of the stimulus is low, then he also couldn't see it. Okay. Now this is another sagittal view of the brain for the human and macaque brain. So the size of the brain is very, very small in the macaque monkeys. And the location of primary visual cortex is in the back of the brain here. And then sort of deep into the middle sagittal kind of plane. And then in the human, there's a little bit of the part of the V1 that is visible from outside. And in the case of monkey, V1 is quite exposed to the back. So here, almost the entire back of the brain, is dedicated to expose the surface of the V1. Okay. And to better understand how it looks like in terms of the blindsight region monkeys, I just prepared for this 3D kind of representation. And here, this is a coronal section slice. So you face to the left, and then this kind of vertical slice is stuck together one by one. And then if you do that, the blindsight monkey's lesion looks like here. There's a hole in the sagittal coronal slice. So if you look at only the left hemisphere of the blindsight monkeys, then this part, this black part is the one that is disconnected. And then that goes from all the way from behind until relatively mid part. And now that basically covers cleanly the V1 of the monkeys. And then just to recap, the V1 lesion monkeys performance and comparable performance of the human blindsight patient. This 2002 paper by Story compares the performance of the monkey Rosie on the left side, and HK, that's another blindsight patient, human, and the right side is again Rosie, and the comparable kind of format of the task. Okay, so Rosie in the forced localization task, then she can actually do above chance up after 30% of the time of the contrast. She can choose where the stimulus is if it's two alternative, right? And then when using the normal hemisphere, the let's say left hemisphere side, she was able to do it, even after 3% or 5% of the contrast. So the contrast performance threshold is very threshold is very low here for the normal hemisphere, but quite high for the blind hemisphere. And then in the detection task, when she was given the third option of saying no, remember last week that we talked about quite a bit about this, bars. When they say, when the Rosie saw nothing, they could actually press the button for no. And then under that condition, this near threshold stimulus of the 4% stimulus, she was able to perform roughly like 50% to 70%. And then same for this HK blindsight participants at the lower contrast. But both Rosie and HK press the button for no, I didn't see it, when the stimulus is presented at the blind field with 99% contrast here and here. Okay, 99% contrast corresponds to this. This means that the Rosie and also HK would have been able to accurately, correctly, localize the stimulus, whether this is here or there. But if they are given a chance to say that, no, I didn't see it, then always they press a button for that, almost, okay? There was a case here, it seems she pressed the button for the presence of the stimuli. Okay, but anyway, almost always they deny seeing things. So it's a quite, reliable replication of the blind sight phenomena in monkey. Now, more modern kind of, blind sight monkey research has been done, and here using the, lesion of the monkey, and without sacrificing monkey, they could now image using a neuroimaging which exact part of the monkey's brain was lesion. So this is very nice because we can make sure that which exact the brain lesions are damaged while monkeys are alive and doing the task and are also recording the neurons and so on. So this is Yoshida's studies, and then monkey T and A's lesion are visualized. And the same monkey was now tested with visually guided suckers. So in this paradigm, monkey are supposed to fixate at the center and then flashed with some kind of stimulus, only one at a time. And then if the monkey is making the eye movement to that location precisely, then they will be rewarded. But they need to do it, rapidly, stimulus, sucker, stimulus, sucker, and so on. That's relatively easy to, train monkeys. And then what Yoshida found was that depending on the location of the stimulus, normal hem field, they make an eye movement really accurately, like this. When you present a stimulus, it's almost always dead on, right? Like this way on the left side. And then blind side affected part, also they are quite accurate actually. And accurate, but there are some kind of mistake as well. That's why you see the spread of the things. But quite interesting thing is that when he compared that mistake pattern between the spread threshold clearly visible versus the near threshold that subject monkey sometimes didn't see versus affected but high contrast, then what you can see here is that in the case of a near threshold, something similar to the masking situation where we sometimes see and we sometimes don't see. And usually we know where when we see it or when we don't. And then you kind of able to see that when this happens, monkey actually make an eye movement here, even though it was presented here, red one. Or monkey make an eye movement here, even though the target was presented here, black one. And this means that when they make a mistake, it's just a complete guess and then randomly make an eye movement to one of the 5 target locations. However, in the case of the blindsight-like situation, they are imprecise compared to the normal threshold. Normal visible side, this becomes much bigger, right? And same here, the precision becomes worse. But they don't make a mistake like this in near threshold situation. There are some small cases where, when the stimulus was presented and then make eye movement here, but roughly speaking, their inaccuracy is just, clustered within the more, correct locations. And that's qualitatively different from the near threshold vision. And this is a further, quantification. And what I'm saying is that, when there is a mistake, there are some mistake that shows, a green presented here and then reporting here, or a green report here. It's rare, but it happens. But in the case of this affected blindsight situation, this kind of thing doesn't happen. It just becomes imprecise, but still, reported more or less accurately. So in sum, the blindsight behaviors are mirrored and reproduced in the B1 lesion monkeys. The nature of the deficits are reliably replicated. Visually guided behaviors, for example, walking or detecting to touch, moving eyes to the target are spared, retained. And the blindsight seems different from masked vision or near threshold vision in normal observers. This is indicated by this precision of the eye movements. So now the question that we started before returns. Is there any clear-cut way to distinguish between conscious versus non-conscious vision? I mean, the eye movement stuff is quite interesting and telling, but it's not super clear, right? So what we want is more clear-cut and better and easier way to distinguish between conscious and non-conscious vision. And that comes in the next video.